Introduce the session. For those that don't know me already, my name is Nigel Osborne. I'm co-founder of Phraktion. For those that aren't so familiar with Phraktion, if there's any sort of external guests this afternoon, just to remind you that Phraktion is really the marketplace for fractional expertise in pharma and med tech. We've grown substantially we're in our first year with a network of over 200 people today. So that's Phraktion. And a warm welcome to our peer-to-peer webinar this afternoon. Just a quick reminder, this session is being recorded. So if you have any issue with that, please kindly dial off. That's your choice, of course. But otherwise, let's get cracking with this session. And the session will be presented by Dr. Nick Moore, and he'll be taking us through drug discovery and development, a project manager's nightmare, apparently. Nick should know this. He has 40 years experience in the drug discovery early development arena. In fact, we can claim he's a substantial international expert in the field and very well recognized for doing that. Actually, just getting to know Nick a little bit in recent days, sort of sharing war stories around really the challenges around drug discovery, and unfortunately, many bricks of the wall fall out as we go through development, and lots of lessons to be learnt about that. And we actually sort of put it in terms of gold prospecting sometimes. You need to dig through a lot of earth to find the golden nuggets. And I think Nick is going to show us a lot of those golden nuggets in the presentation he's about to provide. So without further ado, Nick, over to you to take us through your story.
Okay, well, thank you very much. It's great to talk to you all today. And as Nigel said, I'm supposed to talk about the gold nuggets, but I'll probably be doing a lot more talking about the earth rather than the gold nuggets. But moving on, I thought I'd just give you a bit of background to myself. As Nigel said, I've been playing this game now for 40 years. And my background, I'm from Yorkshire, Humberside. It kept changing its name. But I was actually born in Hull, and I was born a pessimistic cynic. It's really in my DNA. And I think that's actually important in early drug discovery, because every team needs both optimists that believe everything will move smoothly and forward quickly, and then they need the pessimist at the back of the room who puts his hand up and points out that he's not convinced that things will go quite to plan. And I'm pleased to say that I've been right more often than wrong when I've suggested that the plan was too aggressive, too optimistic. But so my background, I'm a neuropharmacologist, did a degree in pharmacology at Sunderland Polytechnic in the days when it was a polytechnic. I got into pharmacology because I couldn't get in anywhere else. In fact, just before I went up to Sunderland, a few days before the course started, I didn't even know what pharmacology was, but did a bit of reading and thought it sounded interesting, so I'd give it a go. So 40 years later, I'm still giving it a go. Did a PhD in neuropharmacology at the University of Birmingham. And then I joined Eli Lilly and Company at the research centre down in Windlesham in Surrey. And while I was working there, I had the great opportunity. I led the preclinical development team that discovered the antipsychotic Zyprexa. In fact, I doubt if they'd have ever let me work on the project if at the time they knew it was going to be a product that was going to have peak sales of $5 billion a year. But I was the first person ever to dose it in animals when it was just a yellow powder called LY17053, and saw it move all the way forward from in the late 80s all the way forward to 1996 when it was launched onto the market in the US and UK, US and Europe. And in fact, that's one of the things I've been most fortunate about, because as I go through this presentation, one of the things people need to be aware of, that in early drug discovery, you can spend your entire career trying to discover a drug that will make it all the way to the marketplace. And the vast majority of early drug discovery research scientists are never fortunate to see a compound go all the way through to the clinic. So I was very fortunate in that respect. Mm-hmm. Then, thanks to a reorganization at Lilly in 2005, I moved to the US for 12 years, and first worked at Lundbeck for 5 years down in New Jersey, then moved to Albany Molecular Research in Albany, New York, Charles River, and then worked in a number of CROs before taking on my current role, where I set up my own company, and now my major clients are a nutraceutical company, Prostasis, and Mycomedical Life Sciences, which is a mushroom-based pharmaceutical virtual biotech company. So moving into the presentation, I'll talk about the early drug discovery process, then give you a case study around an anti-obesity agent that I was involved with, then say a little bit about what can go wrong and what does go wrong, and then a glossary of terms that, in my experience, project managers aren't particularly keen on as they try to put together the plans for how quickly things will move forward. I will then briefly mention the role of AI, because these days no talk is complete without at least mentioning AI once in the talk.
I didn't have to ask you.
We'll end up with some questions, or, well, I'll see if there's any questions. So the early drug discovery process really is a cascade of events, and it starts off with an idea that may come from academia or from internal research. A new target is discovered, and it's thought that maybe if we block that target, or if we stimulate that target, we may well have a beneficial effect in one or more diseases. And normally the target's been associated with a particular disease. And because my background's in CNS, I'll probably talk mainly about CNS targets. So you find the receptor in the brain, develop a selective agent that interacts with that compound, with that target. But obviously, as we all know, there's a lot of things between you and the target. So as well as having high affinity for the target, which is achieved in vitro, in, if you like, the test tube, you've then got to get the compound to the target. So you've got to have the right pharmacokinetics. It's got to get absorbed. It's got to cross the blood-brain barrier. Sufficient of it has got to reach the site. And also it's got to have the specificity that you want, so that you don't have a lot of unnecessary off-target toxicology. And all these things have to come together to have a compound that you can move forward into toxicology and safety. And as I say, at each of those points, as I'll mention at the end, a lot of compounds fail because they don't meet all the criteria. And in fact, this is just an idea of how this is done. So where do the compounds come from originally? Well, particularly these days, there's in silico design. So a computer you'll take the 3D crystal structure of the protein, and then look at how compounds dock with that protein. And obviously on a computer, you can screen millions of compounds to identify a few thousand that you then take into later screening, where you actually make the compounds. And obviously now with different types of AI, not only can you see which compounds bind to the target. You can also rapidly see whether the likelihood of success is high based on what's already out there in the public domain. So those compounds are then synthesized, or a lot of them these days you can buy. So there's companies that just sell libraries of compounds. So they'll sell you a library of a few 100,000 compounds, and you can then put those through your screen. Also, you can have, or a lot of CROs and big companies now have their own libraries, and you simply screen that library, hoping that you'll find a compound that interacts with the target. And then the medicinal chemists can take those hits and start to rationally design them to incorporate the other activities you want that allow them to get into the brain. So it's important to have the in vitro DMPK, pharmacokinetics, and also, as I say, the cross-reactivity that may occur. This is just a typical screening cascade looking at compounds that interact with transporters in the brain. So you have the computational chemists that direct and help medicinal chemists synthesize the compounds. But I would stress that in any good early development team, having good medicinal chemists, particularly medicinal chemists with many years of experience, is very important, because the number of times the in silico, the computer tells you what compounds it thinks you want to make, and a good chemist will turn around and say, well, I tried making that 5 years ago or 10 years ago, and it didn't really do what you wanted and had cross-reactivity, etc. But in the early stages, some of the things you have to think about when planning taking compounds forward, for example, how many compounds can you make per week? How much compound do you need? And what the turnaround time is between making the compound and getting the results back. And one of the questions there, for example, is, with the amount of compound, do you make enough compound in the first synthesis to get you all the way forward through to stage 3 on this diagram, knowing full well that the vast majority of those compounds won't have the desired activity? Or do you make a small amount of the compound to just see whether it has the desired activity, but then to move it forward, you have to remake it, and that adds to the timelines moving forward. And this is often a debate between different people in the group, and the project manager tries to manage how you make this and how you move compounds rapidly forward through the screening cascade. Also, some of the metrics that one uses is, for example, compounds per week synthesized. The issue with that is if you have a milestone to achieve by making X number of compounds, then you tend to focus on the ones that are easy to make rather than the ones that are the quality ones that need to be made. So you actually sometimes end up making a lot of compounds that the chemists actually know may not be good, but it allows them to meet the timelines. So it's somewhat difficult to set, good goals for each stage of the development. So, but once you've got the compound, you then have to, that meets the criteria, you move it forward into assessing the cross-reactivity, the activity at cardiac channels, the hERG channel, and also try to demonstrate target engagement in the brain or the tissue that you believe the compound's going to work in. And at each stage there, a compound will fail one of those. So it may have ideal properties to take forward in terms of interacting with the target, but it may have unwanted effects on other systems. It may have poor absorption. Very often, in my experience, the more interesting the compound, becomes, the less soluble it also becomes, so that it becomes more difficult to work with as you try to move through the screening cascade. But as this is drawn, you can see you identify your compound, you check it's safe, no cross-reactivity, interacts with the target, you easily resynthesize it, and then you put it into additional further testing, particularly in vivo animal testing, to demonstrate the efficacy in models and have the appropriate PK characteristics. The reality is somewhat different to that, and compounds are constantly failing at different stages. But hopefully you will, well, in this, it says 6 months, you'll get to a candidate's decision. But I think this is actually one of those screening cascades that you make at the start of a program, and you tell senior management it'll be 6 months to get to the candidate. In reality, it's probably nearer to a year or just over a year to achieve this, if you achieve it at all. Because one of the other challenges is, when do you give up? When do you think you're not going to achieve the target? You have a lot of interest, time invested in it, but sooner or later you either end up with a candidate or you just have to admit defeat and move on to a different project. This I won't dwell on, but it shows the types of activity. You set typical criteria for the properties of the compound, and basically this is what I've just talked about, that it interacts with the target, has no cross-reactivity, or cross-reactivity at least 10 or 20-fold what you're looking for the target, that the compound has some solubility, it crosses the blood-brain barrier in this case. So you have a pretty good idea what you need to achieve for the compound to become a candidate. But as you can see from this, trying to identify one molecule that incorporates all these different measures and there's the ideal characteristics across all these is actually very difficult. And very often there's a lot of debate about, you know, can you manage with a little bit more of this and a little bit less than of that to be able to move the compound forward. And in fact, I think one of the biggest things that reduced the rate with which compounds pass through the development pathway was the invention of the Excel spreadsheet. Because as you can see on the right-hand side, those criteria are on an Excel spreadsheet. And what I haven't shown here is the compounds are in the next column, and they want to see ticks by every one of these lines. Whereas in reality, some of them come into play and you have to look at them carefully. For example, if you have activity at the hERG channel, you may well produce effects on cardiac function. But if you have a very potent compound and high plasma levels, it may act at the target long before the hERG is a problem. But if you have strict criteria, that compound would fail. And this is just something I've lifted from a publication that was entitled Why do 90% of Compounds Fail in Early Discovery? So if we just focus on the left-hand side, the blue to yellow, and just look at the numbers of compounds that you can see the attrition rate is very high. Once you've identified the target, you then have thousands of compounds. In fact, these days it's much more than greater than 10,000. And slowly it whittles down until you finally get the compound that can go into phase 1. Most projects, you end up with, you know, at the end of the preclinical tox testing, you have the one compound that you're going to take forward, or maybe 2 compounds if you have a backup to the compound. So I'll just show you how this works in real life. And this is an example of a compound taken forward for obesity. This was an MCH1 antagonist. And first of all, this is where the target is. And this is, somebody once called it neuromythology, when you try to pretend you know what is happening in the brain. So the bottom, the blue area is the hypothalamus. And then at the top, you have the higher function in the cortex and the reward centers like the nucleus accumbens. And MCH1 antagonists were thought to work in both the hypothalamus to switch off your appetite and also in the nucleus accumbens to switch off the rewarding properties of food. Because as we all know, in humans, there's a huge reward component rather than just the satiation. So when you go out to the restaurant, you eat the main course, and that's more than enough to keep you alive and stop you starving to death. But then you have a look at the menu, you see the desserts, and then the reward centers in your brain kick in, and you have to have the dessert despite the fact you're completely full. You even say, well, let's give it a minute and then we'll have dessert. Well, you don't really need to do that. I refer to it as the creme brulee effect, because whenever I see creme brulee on the menu, my nucleus accumbens kicks into overdrive. So we went about trying to develop an MCH1 antagonist. And this just shows, after we'd looked, this was a project where we actually synthesized all the compounds to screen them, because there was a lot of prior art in the literature suggesting the types of compound that would work. The problem with them, they all had a lot of cardiac safety issues. So we had to get round those issues. So we identified a lead compound. So in the first bit, this is the physical chemical properties of the compound. So it had a reasonable molecular weight. You don't want the molecular weight to be too high, otherwise it's unlikely to cross the blood-brain barrier. You then have some of the other characteristics that help with solubility, etc. And then in terms of the target, we see that at the MCH1 receptor, it had high affinity for the receptor. It was also a functional antagonist, because sometimes, you're trying to develop something that blocks the target and you actually end up with something that stimulates the target. So you have to confirm that not only does it bind to the target, but it will do what you want, in this case, inhibit the target. Then, having got that, you also want a good separation, or you ideally want compounds that aren't rapidly metabolized. Otherwise, you'll be taking the compound 4 or 5 times a day. So the target for an anti-obesity agent would be something that's safe, or very safe, because, of course, it's not a life-threatening disease in the shorter term. Obviously, some of the comorbidities are an issue, but you need a safe compound with minimal cross-reactivity, and ideally something that can just be taken once a day. So you do the measure the PK, measure the half-life, also look at the protein binding of the compound to again show whether how it binds to the different proteins, because that gives you an idea of how much is actually free to interact with the target. So you may have a lot of compound there, but if it's 99% bound to protein, you've got a lot less available for the interaction with the target. Having got that, you then run it through a whole range of safety profile in vitro. So again, to look for reactive metabolites, the metabolic profile across a range of species, because that will dictate which species you use in the toxicology. So for example, this particular compound had, when you looked at it, there was a metabolite that was seen in humans that wasn't seen in either the rat or the dog. And normally you would use the dog as the second species in toxicology, but we had to go with the non-human primate because that also had the same metabolite. And it's important that not only are you testing the toxicology of the active compound, but you're also testing the compound against any potential metabolites that may well be toxic. So this graph then shows, having got all that, so that's about, probably in the case of this compound, about 6 months work to get to that point, once you've got the compound. So there was probably about a year's work before that to identify that compound. Then you take it in vivo, and this is looking at the effects of the compounds in a model called the diet-induced obese mouse model. So these mice have been put on a high-fat diet. It's often referred to as a Western diet. So it has a diet that's very similar to, sadly, what we all eat. The animals put on a lot of weight. The average mouse is probably normally about 36 to 38 grams at this age. But you see, these mice are about 10 grams heavier. And then you give them the compound, and we see that the green bars, the MCH1 antagonists, produce a nice reduction in body weight over time. And it's much better than the standard that was used here, sibutramine, that has a very rapid effect because the animals just stop eating for some reason, but the weight then slowly comes back. So this is the ideal, looked like the ideal candidate compound. We also here ran the receptor occupancy assays to show that not only did it reduce body weight, but it was doing it at doses that interacted with the receptors. This compound then went into toxicology, and we were able to establish that in the toxicology studies, it had a good margin of safety compared to the efficacy. So the blue area on the left is where the doses are that cause the efficacy, and the doses on the right, the purple line, is where you start to see some toxicology. And you will always see some toxicology with any compound, or the vast majority of compounds, if you push the dose high enough. Somebody once said to me that the difference between pharmacology and toxicology is simply the dose. And in fact, the FDA and other regulators actually like you to show that you've got some sort of signal of toxicology at higher doses so that you can actually measure the margin of safety. So the plan was, as long as you stay within the blue area, then you can dose take the compound forward into man and start the phase 1 studies. And hopefully you will have a compound that is safe and efficacious. So this was by then about 2 years, no, over 2 years, because the tox studies take nearly a year. So about 2.5 years from when this compound was first identified. We took it into man. We looked at it on feeding and appetite in man. And unfortunately, it turned out we had to go to much higher doses in man than was anticipated, than we saw in the mice, which meant that separation between the blue area and the gray area started to overlap. And that meant the margin of safety was very small. That again, you were seeing the QTC prolongation at doses that produced a reduction in body weight. So 3.5 years down the line, the whole thing came to a grinding halt. And for most people in early drug discovery, that is a typical life cycle, and it has its exciting days and its eureka moments. But the vast majority of the time, as Nigel said, you're just shoveling dirt from one side of the lab to the other without finding any nuggets inside it, which then makes it difficult from a planning point of view to put together a neatly drawn Gantt chart that shows how each bit will move forward, what resources you want to bring on board and when, and allocating resources that you then don't need because either things have slowed down or the whole thing has ground to a halt. So here, I just want to say how, AI is going to revolutionize all that, apparently. And if you're a biotech wanting to raise money, you must put AI on the end of your company name, or you probably raise more money if you put AI on the front of your company's name. And it does have great potential and is very useful. But, I'm old enough to remember when I used to go down to the library and actually get a journal off the shelf and look at the paper. And then you'd have to go and get another journal and find the reference from that. Then Professor Google came along and you were able to type into Google the search and you'd get a lot of the papers all would come up at once. Now with AI, not only does Professor Google find the papers, he also reads the papers for you and gives you a summary of what is found, and also potentially the next steps that you may want to think about using. The problems, as I see it with that, is it's only as knowledgeable as the data sets that have gone into it. And sometimes if you look at the literature, there's a lot of information in the literature that isn't really accurate, or it doesn't really mean what it says. And a classic example of that is if it's looked at a press release or some report of a clinical publication. My view is if a press release starts, the compound was safe and well tolerated, that means it didn't work because the likelihood is if it had worked, the first line of the press release would have told you how wonderful the drug was and how it was going to make the biotech millions. But nobody ever says that the target was a bad target, it wasn't druggable or whatever. So AI won't have that background knowledge and look at the nuances of how these things are presented. So it really does depend on the information that's going in. And as I say, it has a difficulty assessing the quality of that information. Also, what it lacks is, the human know-how, the jumping from one thing to another, the creativity of, I know this is a dumb idea, but let me do it anyway. And sometimes those dumb ideas actually work. And as I've mentioned, you shouldn't believe everything you read, particularly academic papers. A lot of them are clearly written for a PhD student or a postdoc to pad out their CV, and the data isn't repeatable, or it was done many years ago. And again, how you filter that is very difficult. So I finally say that, AI, it's a computer-based thing. It lacks the ability to think outside the box, because at the end of the day, it is just a box. And I think, it'll be some time before it really comes into its own the way the press like to talk about it. Although having said that, it's extremely valuable. You just have to be aware of the limitations and use those limitations and carefully. I know an example of an AI-based drug discovery company that, had press releases about the cut down the time to develop the drug to a matter of months compared to years. And then at the end of the day, when the structure was published and the patent came out, one of the medicinal chemists I was working with looked at it and he said, "That's not too surprising." He said, "I could have made that. If somebody came to me and said, I want a drug to interact with target X," he said, "I could have turned that out in a week." But he said, "I also know that target X is a waste of time." But, this particular company has raised millions on the back of that. So just to wrap up, in fact, I'm a bit over time, so I won't dwell on this. I was just going to point out everywhere where, you know, you can plan for the future, you can have your strategy, and then at each of these points, it can all go horribly wrong. As you can probably detecting, the pessimism is now coming out in full flow from me. I'm not an over-optimistic person. But just to wrap up, in my experience, when you do have a project manager on your project, they seem to like things to be fairly black and white. And the things they don't like are terms when they're putting together their plan is words like probably, it may work, that sounds about right, because there isn't the flexibility in a Gantt chart to have the maybe or that's about right, or how long's a piece of string. And then when it gets down to actually saying, oh, we're going to test this next week, the chemist comes back and tells you that the yield was poor and therefore there isn't enough compound. So they'll have to go back to the start, which adds more time to it. Another favorite was we had a chemist that I worked with who was a very good chemist, but whenever you had the project meeting, he was always 2 steps away from the finished product. And this would go on for weeks. So he became known as Johnny 2 Steps because it happened every time. And then obviously hits don't replicate. So again, the plan has to go back to the start again. A favorite of mine that I use is the end of the week or the end of the month. The key to that statement is you never specify which week or month you're talking about. And then the favorite of medicinal chemists is that the next compound will be the best. The next compound will be the one you want to take forward. So you'd never sort of stop hunting for compounds. You always want the next one to work. So just to wrap up, I think, as I said, I've been in this game for 40 years. So I'm one of the guys that sits on the balcony saying to young entrepreneurs that are trying to raise money, just shouting from the balcony, oh, that'll never work. Oh, we tried that back in the 1980s. But the one thing that does irritate me is that people never search before the year 2000. There's a lot of valuable information out there of what's gone before, and a lot of people these days seem to just want to reinvent the wheel. So I hope that's been of interest to people. It was a rapid run through, essentially covered all the key points of 40 years of disasters. But having said that, you know, I was involved with a olanzapine that was successful. So I'm, you know, one of the fortunate people in my career there has been success. And, you know, hopefully we will, as we use new technologies, be able to reduce that attrition rate, to have more successes than failures. But there's a lot of factors that go into that. And I'm not sure that, you know, the young biotech disruptors necessarily will always be able to do that. So with that, I'll wrap up because I realise we've gone a bit over time, and just answer any questions if anybody has any.
So thanks, Nick, for taking us through that journey. And I know to your merit that you've stuck with it, sifted through the dirt and found some nuggets. So congratulations for that. Just one or two questions coming in on the chat. So maybe I'll just go there first and then open up. But Beth was just agreeing with you, Nick, that I think AI plus people equals gold. And really it needs those two things putting together. Question from Paul, actually. So Paul's just asking whether there's any differences. Is big pharma or biotech better at quitting and walking away when no nuggets are found? Who steps away earliest, do you think? Or
I think that's an interesting question, and I think it depends on the pharma company. because, you know, a lot of pharma companies have this mantra that, you know, that they're aiming to fail early. But I think if you fail early and you set the bar too high, then you miss those nuggets because you're just shoveling the earth without sifting through it to find the nugget. So I'm not sure that that's the right approach, but I think many biotechs stick with the project. You know, I was saying to you, Nigel, this morning, when I was in large pharma, we identified we needed 20 projects to be going into toxicology for one project to come out the other side. And then more recently, I was in Cambridge in the US, went into one of the VCs there, and on their lift, their elevator, as they call them, there were 22 company nameplates next to the lift. And I looked at that and I smiled to myself, because that told you of those 22 biotechs, if the large pharma ratio was correct, only one of them would be successful. And if in a biotech you have one project, you know, it's your crown jewels, you know, how do you let go? You know, some founders come from academia and he's going to do drug discovery much easier and quicker than had been done in the past. You know, they won't want to let go, and they'll have raised the money and they'll keep going and they'll keep trying to raise more money rather than give up on the project. And I think that's as much a danger. You know, failing late is very expensive, but at least you've had all the opportunities you could to succeed. Failing early is cheaper, but you may have thrown away your nugget.
Exactly. So just opening up to the viewers. I don't know if you've got any questions out there, audience. There's the chat, but please feel free to speak up.
Just want to echo, Nick, what a brilliant presentation. Really very honest, because it's not often you see failure presented so elegantly.
So another question here, actually. So again, Nick, thanks for the great summary. And I think a huge advantage of AI is the ability to quickly run simulation. And simulation in no time, and applying machine learning principles. It will depend on the data sources, but where do you see AI adding value there?
I think that's exactly where I see it, the way it can pull information together very quickly, and so that you can, you know, look at that information. Because when you think about it, if you do a Google search, you know, it'll come up with a whole list of papers. You've then got to look at those papers, check those papers, and pull together your own sort of summary. AI at the moment can do that very quickly, and then it can do simulations, knowing, for example, with the structures, what has worked in the past and what could be incorporated into it in the future. And it can look at the toxicology to say, well, in the past, when somebody had done this, it hadn't worked. So you can pull it all together. But at the end of the day, you then have to do the human bit and decide that, you know, is this really as accurate as it claims to be? Has it used the best data? And the analogy I use for that is when I watch something like, you know, a scientific program documentary on the television, you know, it's on astrophysics. And by the end of it, I walk away knowing that I'm now an astrophysicist. Then the next week, there's a program about drug discovery. And you watch that and you think, what a load of rubbish. You know, they really don't know what they're talking about. And then you think to yourself, maybe I'm not such a good astrophysicist as well, because if they've applied the same simplistic approach, then there may be a problem there. And I think AI is the same, that it can generate a lot of useful information, but you have to carefully look at it. I did a search on ChatGPT. I asked it about, I'm now working in the psychedelic medicine space. I asked it for the metabolite of psilocin, which is the metabolite of psilocybin. And it came up with another compound that wasn't the metabolite. But if you just asked it that, you could have walked away thinking psilocin was the metabolite. So I told it, no, it's not. And it's quite good. It does apologize. And it actually said, I'm sorry I got that wrong. I have now rechecked and it's cysteine. So I typed again, no, it's not. And it came back and said, I'm sorry, I've got that wrong. And it said, it's for methyltryptamine. And I said, no, it's not. And it stopped talking to me. So, you do have to be careful and assess the information, but it will pull together that information in a lot quicker time than if you had to do it the old-fashioned way.
Yeah, thanks, Nick. So, Beth, I don't know if you're on the line. I know this is also your sweet spot. You have a question here, but I don't know if you want to come on online and ask.
Sure, thanks so much. So related, because I'm on the data and AI side, I just love data. I can't tell you how much I love data. So,
I should have checked that anybody was working in AI before I rubbished it.
There's some alternative cynics out there.
Yeah, but I agree 100% that it needs data plus people. And I think anybody who's in this space will always say that both the robots need to check the people and the people need to check the robots. It goes both ways. But related to being able to kind of harness the collective knowledge that you're talking about, right? The experience, the deep expertise in this area. Are you familiar with the term metadata?
Yeah.
So do you think that if we could capture the appropriate metadata that kind of allows people to make those connections about the ahas across their experiences and what their findings and these nuggets, that we potentially could create repositories where that knowledge can then be applied so that you can set it aside, knowing that you have something that's continually checking, would this be interesting later?
No, I think, that's definitely a possibility. I think, as these things advance, there will be increasingly the opportunities to collect more data and more meaningful data. As I say, having moved all the way from reading pages in the library to online searching to AI, I see that as being a logical progression, is how you capture that experience and that creativity that is so difficult to for a machine to capture. How does it suddenly just think, no, I'll just try this for the hell of it? And you try it for the hell of it and it works, and you have a
I'm
success.
just laughing because in your story, my very first job was to collect the health of the United States, and I spent months in the basement of libraries, repositories, moving stacks to pull pamphlets out and making photocopies, which I then had to type into databases. So I'm totally with you on that whole journey. But it's amazing if we can begin to capture the knowledge that you bring, that all of us collectively bring, we can begin to actually think about doing simulations that can create some of that magic that the humans are bringing. That just means that we evolve the relationship, not that it goes away.
Yeah, but I think also, as you say, it's how to capture that. And
100%.
Yeah, back in the day when I first started out, it was in the days when you could have a drink at lunchtime. And
yeah, I think I'm as old as you.
Yeah, there was a pub across the road that was just behind where our library was, and there was a gate in the security fence. So you could say that you were going to the library and spend the afternoon, but you actually went to the pub first. But having said that, very often some of the best ideas came out, you know, while you were all just sat around having a drink rather than in a formal conference room, because you were happier to come out with those crazy ideas. And, you know, if you could remember them when you got back, it was a real opportunity. And it's, you know, how do you get your computer drunk, I suppose, is the key question, to be able to capture those.
Oh, they're working on that.
Well, I've certainly poured drink over my computer and it didn't do it any good at all.
Thank you. Great presentation.
Thanks for Beth for coming on. So I was going to say, next you'll be telling me you were smoking in the laboratory next, Nick.
No, to be fair, I've never smoked, but I have been known in the very early days. I had a rat in one pocket of my lab coat as I walked down the corridor, and I had my lunch in the other pocket. The problem was you had to remember which pocket was for rats and which pocket was for sandwiches. For some reason, that sort of thing isn't allowed these days. You have to have a face mask, you have to have glasses on, and
I think I think we've all learned more than we'd anticipated during this discussion, Nick and everyone. So I think yet again showing a very fine example of the deep scientific expertise amongst the group, amongst many other credentials as well. So I think that highlights very nicely again the power of Phraktion's network and the interaction and the value we can actually offer to those companies that are sifting through the dirt and thinking about moving into clinical development and beyond into commercialization. So, you know, that's very key. I think that comes off very strongly again in this interaction. So thank you, Nick, for a tremendous presentation and audience participation, for all our partners and any external members today. So thank you for that. We have more webinars coming up over the next week, so look out for those, please. And absolutely, tremendous interactions today. If you'd like a webinar slot yourself, just reach out to me after this. If you want to form groups of people to have a discussion panel around whatever subject you'd like to discuss, then again, let's speak about those ideas. Let's create a webinar around that. So really, thanks again, Nick, and we'll see you all very soon, I hope.
Yeah, and I'd just like to add, so if any of the partners do come across a client that's looking for a pessimistic cynic or a cynical pessimist, then I'll be the one that's available.
Then join the queue, Nick. Absolutely. Thank you, Nick.
Thanks, Nick.
Okay, thanks very much.